particular specialty is, and that's the immune system. And this is the adaptive part of the immune system, what I would consider immunity. So the adaptive defenses, the adaptive immune system, is very specific. It can eliminate almost any pathogen or abnormal cell in the body, at least pathogen. It's in addition to that, it helps to amplify the inflammatory response, so it makes our defenses better. It activates complement, which helps make our body defenses better. And the real shortcoming, the real reason we can't have just our adaptive defense is that it has to be primed the first time. We have to get to know our enemy first for specific foreign substances, and that process takes time. Characteristically, it is very specific. It recognizes target-specific antigens and even parts of antigens, as we'll get to. It's systemic. It is not just going to act at the local site. Antibodies will go to all your body fluids. T cells will search out your entire body. It has memory, which is really different than our non-adaptive defenses. It mounts a stronger attack against antigens it's already seen before. And there are two basic branches to the adaptive immune or the immune system. There's the humoral or antibody-mediated immunity, cellular or cell-mediated immunity. Humoral immunity is the product of antibodies. Those antibodies are produced by lymphocytes. The lymphocytes that do that are the B lymphocytes. Those antibodies, for the most part, are released by plasma cells, which are mature B lymphocytes, and circulate freely in our body fluids. They will bind to target cells, and that binding has multiple effects, but it can, in some cases, inactivate a target if it's a toxin, for instance. And in other cases, it can mark the targets for phagocytosis or by attaching complement to it. So it acts as an opsonizing agent as well. Humoral immunity has extracellular targets. Those antibodies need to bind to things that are outside or on the surface of cells. Cellular immunity, the lymphocytes act directly against a target cell. They kill the infected cells, and they also work indirectly by releasing things that help the inflammatory response and help the immune response to really get going. We're going to have a group of cells, T cells, we're going to call helper T cells. Cellular immunity has a cellular target, so lymphocytes have to actually bind to their target cell in order to do their work. All right, so I've used the word antigen a couple of times. We've actually used it throughout the year a couple of times. So what do we mean by an antigen? An antigen is something that can get these adaptive cells going and provide an immune response, either a T cell or a B cell response, most generally both. These are the targets of the receptors of our lymphocytes, either antibodies or T cell receptors. Most of these molecules are large, complex, and not found in our own body, which makes them non-self. Antigens can be complete antigens or, in some cases, haptens, which means they are incomplete and they have to bind to something else to get an immune response going. But in those antigens, there are numerous antigenic determinants. And those antigenic determinants are the actual sequences of things that antibodies or T cell receptors interact with. So an antigen is going to activate many B cells and T cells. It is possible that antigens can also be self-antigens. We have all kinds of antigens that in someone else would cause an immune response. In us, they do not, because we recognize them as our own antigens. So complete versus incomplete. Complete antigens have two important properties. They are immunogenic and they are reactive. So immunogenicity means they have the ability to stimulate the proliferation of a specific lymphocyte class. Could be a clone of B cells, could be a clone of T cells. There are these cells against this immunogen. Reactivity means that they react with activated lymphocytes and the antibodies are released as a result of this interaction. So foreign proteins, very good complete antigens. Some polysaccharides are very good complete antigens. Some lipids and some nucleic acids. However, nucleic acids are tough because they're, first of all, intracellular most of the time, and there are only 4 different nucleotides in all different forms of life on the planet. Incomplete antigens are also called haptens, and they are molecules that don't start an immune response on their own. They're too small by themselves. Small peptides, some nucleotides, some hormones. However, the problem happens when a hapten attaches to one of our self proteins, and that combination of our self protein and the hapten is seen as foreign. That can cause the immune system to mount an attack because it is attacking the self proteins as well as the hapten. Some examples of this include poison ivy, which most of the population of the world is allergic to, not everyone. Animal dandruff, some detergents, some cosmetics, people that are sensitive to these things. So on a single antigen, which is a part of hopefully a pathogen, there are numerous antigenic determinants. They are the parts of that antigen that an antibody or T cell receptor will bind to and cause a response. Most naturally occurring antigens have determinants that mobilize several different lymphocyte populations. So you're not just going to make one antibody against the pathogen, you're going to make numerous antibodies against the pathogen. And most of them are going to cause different kinds of antibodies to be made against them. We're going to see there are some different forms of antibodies. There's IgM, IgA, IgG, and IgE in particular. So most naturally occurring antigens will invoke all of those, and all those have different individual properties that we will get into. Large chemically simple molecules like plastics tend to have little or no immunogenicity. They don't have antigenic determinants. They're not different enough. So we have an antigen here. Maybe that antigen is the chickenpox virus, and it has antigenic determinant site A, antigenic determinant site B, and antigenic determinant site C. It will invoke the production of at least 3 different clones of B cells, producing antibody A, antibody B, and antibody C. Self-antigens, our cells are covered with the same kind of proteins as everyone else's cells and as pathogen cells in some cases, and we call those self-antigens. One set of important self-antigen proteins are a group called the MHC proteins. The MHC stands for Major Histocompatibility Complex. They're coded for genes of the major histocompatibility complex. And because of rearranging in the number of genes that are out there, every individual has a different MHC set of genes and therefore different MHC set of proteins. Those proteins are going to be important in getting T cells to work, and the MHC compatibility is what people look for when you try to match a transplant and a donor, a patient and a donor in transplant. Okay, the adaptive immune system involves 3 types of cells. I said 2 types of lymphocytes. The ones that produce antigenic responses are B cells and T cells. But B cells and T cells do not work very well if they do not have antigen-presenting cells. Antigen-presenting cells, or APCs, don't respond to specific antigens. They're part of the innate response we saw before, but they do play an essential role in getting immunity started and going. So lymphocytes develop, mature, and activate in order to do their jobs. T and B lymphocytes have a common development stage. The 5 general steps for them are to start in the bone marrow, red bone marrow. The multipotent blood stem cell will go down the lymphocyte line. They will mature. Lymphocytes are educated as to self or non-self in the primary lymphoid organs. B cells remain in the bone marrow and receive their education. T cells go to the thymus to get their education. They have to be educated because, number one, they want to become immunocompetent. So, to be immunocompetent, a lymphocyte, before it's ever seen an antigen, has to be able to recognize only one antigen. They will each display on their surface one unique antigen receptor when they are mature. That's part of the education process. They also have to be self-tolerant. They cannot recognize self-antigens. If they recognize self-antigens, we want to get rid of them because that would lead to autoimmunity. They would then go to secondary lymphoid organs and into the circulation. So they are now immunocompetent. They are able to respond to an antigen, but they are naive because they have not seen their antigen yet. They will leave the primary lymphoid organs, either the bone marrow or the thymus, and seed the secondary lymphoid organs, which we talked about before the break, lymph nodes, spleen, and that increases their chance of encountering the antigen they are specific for. When they encounter their antigen, they will become activated. So a naive lymphocyte's first encounter triggers that lymphocyte to develop further. That lymphocyte is selected. It differentiates into an active cell binding to its specific antigen, and this process is called clonal selection. So you're selecting the clone of B cells or T cells that responds to a specific antigenic determinant on an antigen it sees. If those signals are present, the lymphocyte completes its differentiation into an active cell. And then that active cell will proliferate, make an army of exact copies of itself, which we still call clones. Most of those clones are going to become effector cells that produce whatever is needed to fight that particular infection. But a few of them are going to remain as memory cells, and those memory cells are going to be able to respond to that same antigenic determinant, that same antigen, more quickly the second time it's encountered. B and T memory cells and effector T cells are constantly circulating. Now, the diversity of the receptors, the ability to bind to specific antigenic receptors, is a product of our own genetics. It is not determined by the antigens we see. It's determined by the antibodies and T cell receptors we can make using the variety of genetic genes that we have. And there are about 25,000 different genes coding up for about to 1 billion different types of lymphocyte antigen receptors. There's a huge variety of receptors. This is an unusual genetic situation because gene segments are shuffled around, moved around. We get a lot of variability. This is not your typical one gene, one enzyme relationship. T cells mature in the thymus, and they are under both negative and positive selection pressures. The positive selection pressure is going to select T cells that are able to recognize self-MHC, so they know that their antigen-presenting cell belongs to them, but not recognize it too well. Otherwise, they would go to negative selection. And negative selection, if you recognize a self-antigen, an MHC, you would eliminate that clone because it would be an autoreactive. the active clone. So we need to be able to see self, but not too strongly, not see self too strongly. So we select for those that can see self, but not strong, and those that negatively select for those that do see self too strongly. And that gives us this idea of self-tolerance. And that happens within the thymus.